Before we dive into the heart of the course and the data mining techniques, it's important to talk a little bit about pre-mining your data before you perform any analysis. This is a fundamental step, sometimes called quality control, that is absolutely crucial in any data mining exercise, and it's often overlooked. So there's several reasons why we shouldn't just dive in. The data should have a chance to speak for itself prior to any formal analysis, and you may not always like what it has to say. In fact, typically, I should say ideally, there is a statistician on board in the design of any experiments or data collection efforts. Nonetheless, technology can get in the way. People's interpretation of how the experiment should be carried out can get in the way. There could be processing errors, things that are unanticipated, even with the best design, and they have to be searched for in the raw data and accounted for, if possible, in any downstream analysis. So, data exploration can draw attention to a number of factors. I list only a few here. Any quirks and errors that are in the data, any obvious trends that are either anticipated or unanticipated should be explored. If there are technical artifacts, for example, in the sequence in which the data is collected, you wouldn't want something like that to dominate the hypothesis you are exploring. And these things and just exploring the raw data may in fact direct you in the types of analysis that can be performed. We're going to be talking about a wealth of methods, data mining techniques, and supervised learning, but you'll see that there's kind of more than one way to skin a cat. There's several different avenues you can go down with data mining, and ultimately they should be guided by the hypotheses of the investigator and also what the data is telling you in this pre-processing step. There's this famous saying called garbage in equals garbage out, and if you put junk into a data mining algorithm, it's going to give you something back. I mean, that's what it does. It can process anything and give something back to you. And how much faith you have really should be a function of how clean the data is. So it's important to understand the experimental design and conditions of the experiment. You should try to collect all of the information from the investigators, even if it doesn't seem relevant at the time, because there might be some technical artifacts in your data and so forth. And you're going to want to clean the data, and we'll talk a little bit about that, before you go ahead and apply techniques. And you should be skeptical. The best data miners are the most skeptical. So, how can we start to expose some of these features of a data set? This is an old set of techniques, we'll call it, exploratory data analysis, and it refers to the use of graphs and display to help understand data and reveal trends in the data and so forth. Centers around quick and dirty methods using pencil and paper. Like I said, it's an old technique, but it's still very relevant. And the purpose is to visualize and examine data sets, and it's focused mostly around small data sets. At the time this was developed, we didn't have the types of data sets that we have now. Now, times have changed, and there are, these data sets are on the order of billions. And the size, the space is an issue too. So, for example, billions of transactions per year in financial sectors, social networks that consist of billions of nodes, billions of connections, and covariates that can't even be quantified. You have the Human Genome Project, where you have gigabytes of genetic information. And in astronomy, we have even more. We have terabytes and petabytes of data. So now, data mining, which is really an evolving concept, it evolves to accommodate, new sizes, the increase in size and complexity of the data. We also have to think about it in terms of storage. That's a more fundamental problem. And so these exploratory techniques, many of them will not translate well to to these larger data sets. We're going to be dealing with large data sets in this course. They're not going to be as large as this, but they should convey some of the obstacles that come along with analyzing large data sets. Let's focus at the most simple level first, at a sample or just a few measurements. Histograms and density plots, these are just critical to get an idea of the distribution. So here I've plotted this is total length for some, I'm not sure, I think it's from a possum data set, so the possum length. And we can see that if I plot the histogram with this bin range, that it looks asymmetric, and here I look like I have a normal distribution. And then you can plot the density curves right over there, and I'll show you how to do histogram plots and so forth. So this is something that I typically do at one of the very first steps. Another EDA technique, which you saw on the cover of that book, is stem and leaf plots. So these are heights for 37 rowers, and I'm going to show you how to do all of these things in R, by the way. You can see that this is just another way of viewing the distribution and how this may be impractical on a large scale as well. You can see the median, lower quartile I have flagged, and the upper quartile I have flagged. And you can see that there's probably an outlier here. Okay. Box plots, these are very useful. Here I just have one type of measurement being plotted here. I have the median, the lower quartile, upper quartile. These whiskers can mean different things. Sometimes they cover the whole range of values. In this instance, it's 1.5 IQR, and we can see that there is an outlier here that we might want to get rid of in our data set. So that's great for outlier detection, and I'll show you how to do this type of plot, as well as if you have different measurements, how to do a strip box plot, which is useful for seeing how these measurements sit relative to each other. Okay. Time series data. We probably won't deal too much with this. We may have a couple of data sets like this. Here I have years. This is London measles deaths. So these are all deaths per year in a population on a log scale. And you can kind of see this saw-like pattern. It's kind of difficult to know what's going on. You have a lot of missing data here too. And these dots up here, they stand for the overall population, so that data is sparse too. It's not available back here. It's hard to know what's going on, so I just clipped it and zoomed in on this region where I have a full data. And now we can get a better idea of how severe the deaths are and due to measles relative to the population. And now I've also rescaled the y-axis. So sometimes it's just a matter of looking at the data, the overall trend, identifying a region you want to zoom in and picking that to see what's going on. Here we have just a scatter plot. This is milk sweetness. So these are all people who have rated the sweetness of two types of milk sample 1 and milk sample 4. And you can see that, there's a positive correlation here. And I've also made these little hash marks down here to give, the exact, the exact value that they chose for the range of sweetness. And that's useful too. So this is just a basic scatter plot. This can be done in more high dimensions too, and you'll have to do that in your homework. Here we have some data that has to do with electric resistance in juice, which is different. So you can see that there's definitely a negative correlation here, but it doesn't look linear. I fit a lowest locally weighted scatterplot smoothing curve to this data, and it represents it pretty well. So now we're just looking for overall trends. It's very useful. Let's look at this data. This is brain and body weight. And I'm sorry, I don't know the units for this, but this is the raw data. And I look at it and I say, I don't know what's going on here. There doesn't seem to be any relation because of the scale. It's very typical to have to do a rescaling. If I put this on the log scale, I see that there's an obvious strong correlation between body and brain weight, which is probably what we would expect. This is called a strip plot, and this is nothing more than just plotting them. This is the length of an egg, and these are different egg types. And we can kind of get an idea of where the egg types sit as far as size or weight by just looking at these, but we might want to summarize them in a box plot fashion. So now here are the box plots again that I showed you, and we can see where they sit relative to each other. So this egg length is probably much smaller than this one. Here's a density plot, nothing more than what I did before, but now we have two different, I'm not sure what this is, measurements of ear for possums, I believe. It's not really important. The important part is I've broken it out here by female and male possums, and these are the two traits, and we can see that there's a big difference. So we can break these out by covariate. We can plot these density plots and also plot the curve or the points themselves underneath. So you still kind of retain that histogram information. It's a nice way of doing that. And we can see if we weren't to plot these points, we might suspect, what is this over here? Is this some kind of evidence for a bimodal distribution? But we see it's driven by one outlier, and we might want to get rid of that in our analysis. So here, this is broken out by female. Here's the density. Now I'm going to break it out even further, but just use scatter plots, broken down by multiple factors. So here I have age, older and younger, so a categorical age, and we have sex, female and male, and I'm just doing the plots here of this trait, CSOA, which has something to do with car tint. It's not really important. The point is, I can see in the scatter plot, what's going on with the trends, and I can go one step further, and I can fit some smooth lines over those values to get a better idea of any trends that might be there. Now, as I said, I'm going to show you how to do all of these things in R. This is just a taste of different techniques that you can use to visualize and detect things in your data. It's by no means comprehensive. The analyst should look for things like outliers, any missing data, clusters in the data that may be there, unexpected patterns within groups, between group differences that are obvious in the scatter, or obvious can be detected with the box plots, and whether or not there are unanticipated trends associated with the data. For example, with the order of data collection, or there's some confounding in the treatments, you'll want to look for those kind of things. And that's going to be more experiment specific. So collect all of that information from the investigator. Now, this is just to kind of drive home the point that there's a lot of work that has to be done before data mining is even approached. So here I have written here a basic list for knowledge discovery in databases. This is kind of the workflow for what you would do if you were given a data set. The first thing is select the target data set, and you may be given the data set, but you want to know, you may have to trim it down, especially in the large scale problems. Which data should you use? Which variables and cases are to be used? So if you have 38,000 gene expression traits and you want to build a classifier to predict prostate cancer, 38,000 might be a little bit too much. You have to figure out a way to zoom in on particular variables and any cases that you want to admit or eliminate. Data cleaning. Once you've selected the data set and you've zoomed in on the variables, you're going to have to remove noise, any noise that's in the data, identify outliers, and impute missing data if appropriate, etc. Pre-processing of the data. So once you do that, you'll decide upon the transformation. For example, if you have to do a log transformation, you may actually do this before the cleaning or in conjunction with the cleaning. You want to track any time-dependent information and any processing information. You'll want to collect that as well. Decide which data mining technique is appropriate. So once you've done all of these things, you should have an idea of what the raw data is telling you. You should also have an idea of what the investigator is trying to show, and from that you may decide what technique is appropriate. So this is a whole lot of work, can take on the order of months, and sometimes longer. And only then can you approach step 5, which is what we're going to talk about for the rest of the semester. Analyze the clean data using the data mining technique that you've selected. And once you do that, you'll interpret the results and assess any knowledge derived from the data mining process. So it's a long list of things. For the rest of the semester, we're going to be talking about 5, basically. And but 1 to 4 are crucially important, and like I said, they're often overlooked. Here's an example in my own field. This is a paper that was published in a high-profile journal, and what we have here are a bunch of metabolites. This was an experiment done in mice, and this is just a correlation map. So if you think about, if you're going to do a correlation matrix, we have, there might be 50 different metabolites measured. And what we see here, and it's clustered. So, we haven't talked about cluster, but probably you know what the idea is. So there's a lot of similarity here, right? Oh, you might wonder, wow, this is what we call a module. What's going on here? These are all of the amino acids, okay? And let's take it a little bit further and look at what's controlling these amino acids in the genome. Here they are, and we can see that their patterns, we call it QTL patterns, are very similar. They all have these hot spots. They all have the same hot spots, the genomic hot spots. So there are, spots on chromosome 8 and 9 that are controlling this whole bunch of metabolites. And, from a biologist's perspective, this might seem very interesting. And so this is kind of the pre-processing. You do a little bit of correlation study. If I saw this, I might think it's a red flag. How can all of these amino acids be so highly correlated? I might be hesitant to put too much confidence into this similarity in QTL hotspots. But this was done, and they went even further and built a graphical model for predictive purposes to see, if you change genotypes here, what are the downstream consequences. So now we have not only a full analysis being done, but we have a graphical model for prediction. And as it turns out, the way that these are measured, they're so correlated. They're not independent traits, although they are considered independent in the analysis. So this is a big problem, and one that can be easily detected just by talking to an investigator, looking at correlation matrix and saying, oh, the way that these are measured, they're not really independent. They shouldn't be considered independent. But in fact they are, and that's the theme of this paper. So there's a lot of things like this that stick out in the literature. Like I said, this is a high impact journal. And so, as a reader and as a statistician, even though you may not be a statistician, you should be skeptical of the methodologies that are applied and the data underlying the methods. And when given your own chance to analyze data, you just be very, very careful. Not only in the cleaning process, the data mining techniques that you select, and the interpretation, not to overinterpret the analysis. Okay, so I will cover in the next segment some of the basic R commands and introduction to R to produce some of the graphs that we talked about. Thank you.